Thank you very much. So this brings our opening remarks to an end, but now the fun is going to begin. So it is my pleasure now to look for Paul Zizek, who's down here, who's going to be the chair of the first session this morning. So I'll let Paul take it on from here.
Yes. Okay. Thanks, Karim. I will just get right to it then. I'll introduce our first speaker. We have a pleasure to have Joel Zylberberg here from Toronto. Joel received his PhD in physics from Berkeley in 2012. And then he was assistant professor of applied mathematics at the University of Washington, and then assistant professor of physiology and biophysics in Colorado, the University of Colorado. In 2019, he moved his lab to Canada, to York University in Toronto, where he is the Associate Fellow of Learning and Machines, and where he's a Canada Research Chair in Computational Neuroscience at York University. And he's also an Associate Fellow of Learning and Machines and Brains at CFAR and faculty affiliate at the Vector Institute for AI. So his research really lies at the intersection of neuroscience and AI, and he's exactly what this conference and this unique group is about. So he's received multiple rewards, including Google Faculty Research Award in Computational Neuroscience, a Sloan Foundation Research Fellowship, and the CFAR Azrieli Global Scholar Award. And so his talk is entitled "New Biorealistic Artificial Neuron Types May Help Us Develop More Human-like AI." And so please join me in welcoming Joel Zylberberg.
Perfect. All right. Well, thanks very much, Paul, for that kind introduction. And thanks to the organizers, Karim, and the rest of the group that put together this great meeting. You know, if you look around the world right now, there really is nowhere that has a more active and vibrant community in neuroscience and AI than Montreal. And I think that the topic of this meeting really reflects that. And so it's a real treat for me to get to come here and engage with all of you on this topic, which really forms my life's work. So with that, we'll just get started. You know, as Paul mentioned, the topic of my talk is this idea of somehow trying to use neuroscience data and neuroscience models to make better machine learning systems for AI applications. And so I'll start by pointing out that if we think about the way we build deep learning models now for AI applications, there's really a few knobs we can tweak in order to determine what our models will do. Right? We get to pick an architecture, an objective function, some learning rules, usually backprop or some variant of it, get to pick some training data. And my talk will really focus on these three of these items highlighted in blue, which is architectures, objective functions, and training data. And the claim that I want to make, that I'll try to support with some examples from my work, is that if we want to make brain-like or human-like AI, the right choices of these things in blue are the ones that lead to models that can predict brain activity under diverse conditions. If they're able to do that, then we can sort of add readout layers that will convert the simulated brain activity into decisions or actions, and as a result have systems that we know are implementing human-like or brain-like AI because they are doing it, you know, using the same kinds of representations that our brains use. And so I'll talk about how to build systems like this, or how my lab goes about building systems like this, and I'll describe two specific lines of work. The first of which is that we directly use brain activity data as a regularizer, or in some form as like a representation teacher, to show neural nets how they should represent input information and use it to perform tasks. The second branch of research that I'll talk about is one where we directly build in new artificial neural network layers that contain neuron models that are fully trainable. And as a result, we make deep learning algorithms that operate, you know, in the way the nervous system does. And I'll start with this idea of the brain as a representation teacher. Okay. So sort of motivating this work, when we started this back in about 2016, it was really my grad student at the time, Callie Federer, And this was back when I was in Colorado. And in fact, back when I think when Barack Obama might have still been president. Anyway, it was a while ago. And this paper came out from Dan Yeamans and Jim DiCarlo that really, really to me, kind of opened my eyes to this idea that brain-like models might be good for something besides just neuroscience. And of course, the neuroscience side is very important. Right. So what this plot shows is a whole bunch of different deep learning models that Dan Yeamans had assembled. And on the vertical axis here is how well those models predict the activity of neurons in inferotemporal cortex of monkey. So in the visual sort of object or recognition part of the brain. The horizontal axis here is how good each deep learning model is at object recognition on an object categorization task. And the main point of this paper is that the data kind of slopes upwards. In other words, the models that are better at doing the task are also more similar to the monkey brain. And so Callie and I were reading this paper and had this idea that maybe, if we push further up into the brain-like direction, so we'll go up on this plot, we might also go further to the right. And I should mention that this idea also grew out of a really strong collaboration with Alana Fish, professor at UAlberta, who's been an active collaborator on this and deserves also a lot of credit for initiating this line of work. Okay, so that's our idea. We want to push up on the brain similarity to improve performance on some axis. And so we started by first asking, can we push deep learning models up on that brain similarity axis at all? If we can't, then we'd better stop and figure out how to do that first. So we started by trying to just take in images and predict the activities of neurons in the monkey visual cortex. So in this case, these are data recorded in Adam Cohn's lab at the Albert Einstein College of Medicine. They've got a Utah array implanted in monkey V1. And then they show the monkey a bunch of pictures, and we get the activities of around 100 neurons at a time in response to all of those pictures. We then took those same pictures that the monkey saw, trained our deep CNN to take in the pictures and predict the firing rates of the neurons in V1. And we found that worked fairly well. So of all the different kinds of models that we attempted to construct, the best in terms of how well they can predict the activity of neurons in monkey V1 happens to be this CNN with two hidden layers in it. So that was encouraging to us, telling us that we were able to make models that can predict visual cortical activity. And then we decided then to turn to this other branch of our project, where we said, all right, now that we know that we can predict neuron activity, let's use that property to try to make neural nets that will be better at computer vision tasks. And that's the topic of this paper from Neural Networks in 2020. So the idea here is that we're simultaneously training our neural nets to do two things. The first of which is to take in pictures and return category labels, so the sort of classic object recognition task. I think we were working on CFAR 100 data set for this paper. The second thing our neural nets need to do is to form internal representations that mimic the way the monkey brain represents images. So to do that, we've got the following loss function. I'll explain this in English in addition to in math in a moment. But this is the loss function. The first term here is the difference between the monkey brain representation similarity matrix. So this is an object that I'll describe in a moment, but it's an object that tells you the structure of the representation of images in the monkey brain. This other RSM here is the representation similarity matrix formed by the artificial neural net at any given moment during training. And the first term in the loss function is just the squared difference between those summed over all the matrix elements. So this really quantifies how different the neural net's representation of images is from the monkey brain. And of course, minimizing that loss means that the monkey brain representation is similar to the neural net representation. The second term is the categorical cross entropy that you've seen a lot. This is just minimizing this forces the neural net to return category labels that are similar to the teacher signal in the data set. All right, so minimizing simultaneously these two things forces the neural net to represent images the way the monkey brain does and to categorize images. So what are these RSMs? Well, the idea is for every pair of images that goes into the neural net or that the monkey sees, we can calculate, the activity vectors of either, the neural net unit activation. So just literally the vector of activities of artificial neurons in some layer of the neural net. Or for the monkey, we can take the vectors of firing rates from the neuroscience experiment that were measured when the monkey looked at that same picture. We can then take the cosine similarity between the pairs of those vectors. For the monkey, the cosine similarity between the pairs of vectors for every pair of images, that gives us the monkey representation similarity matrix that tells you, as the name implies, how similarly the monkey brain represents each of those image pairs. And likewise, we can do the same RSM, calculate that matrix for the artificial neural net and get the artificial neural net's representation similarity matrix. Okay, so a lot of detail there. Overarching kind of big picture is our loss function has two things in it: make the neural net representation match the monkey, and do the categorization object recognition task. And the exciting thing for us is that once we do this, this training, we've got a chance to titrate how strongly the neural net is forced to match the monkey brain. That's what this R is here, which is roughly the same thing, although not quite exactly the same thing as this lambda coefficient here that tells you how strongly the neural net needs to weight each of these two factors in the loss function. So we can titrate by changing this number. We can titrate how strongly the neural net tries to match the monkey brain. Over here would be a lot, and down here would be not at all for R equals 0. And then we can see how the object recognition performance changes as we change how much we force the neural net to mimic the monkey brain. And to us, the exciting result is that the peak of this curve is not at 0. In other words, the best neural net that we could make with this procedure is one that is in fact forced to match the monkey brain object representation to a certain extent. Okay. The next thing we asked is if in addition to just increasing the object recognition performance, are there any other benefits that neural nets derive from using the monkey brain as a representation teacher? And we thought that in particular, something like dealing with corrupted data or dealing with ambiguous instructions might be a property that the brain could really help impart on the artificial neural nets. And so what we did is we trained our neural nets that either do not match the monkey brain at all, that's R equals 0, or that do have that two-term loss that forces them to match the monkey brain, that's in red. We took those neural nets and we retrained them a bunch of times with data sets where we had randomly corrupted a bunch of the labels. So during the training procedure here, 10% of the labels are randomly permuted so that a picture of a truck would have a label of suspension bridge on it, or some other nonsensical label. And after training on these corrupted data sets with different degrees of corruption, we asked how good the neural nets were at categorizing images. And we find that the neural nets that are forced to mimic the monkey brain's image representation in red are more robust against corrupted training data. In other words, they can learn to do the task, even if some of the labels they're learning from are wrong. And that is a property that is better than the neural nets that do not match the monkey brain in black. Now, since we did this early study, I guess the preprint on our paper, the first one that came out in 2018 or 2019, and the journal article in early 2020, since then a bunch of other groups have followed this up with other studies that are regularizing neural nets with brain data, largely seeing, and these three in particular, largely seeing that you get increases in robustness from using the brain as a representation teacher, as you might expect. I'll point out one recent study from my lab and again from collaborator Alana Fish. In particular, what we've done is we've came up with a more flexible way to match the neural net image representation to the monkey brain using something called deep canonical correlations analysis. For lots of technical reasons, we think this is a much better version of the monkey brain regularizer than the RSM that we started with. And that's borne out by the observations in this paper that show that we get a little bit better performance using this new deep CCA version of the regularizer. Okay. And where we're going with this, I say this is sort of an opportunity, but we're actively pursuing this now. Alana and I, thanks to a New Frontiers and Research Fund grant from the Canadian government, where we think there's a real opportunity here is that because we can use brain data to help train neural nets to do tasks, we can also pick the brains from which we sample that data. And it means that if we're trying to make neural nets to perform a task in which there is sort of rare specialized human expertise, well, if we can have brain data collected from those experts, then we have a chance to use the expert brains to train the neural nets how to mimic that expertise. So what we're doing now, and I should say we're starting with a bird watching task because it's easier to find experts. And then the next version of this is going to involve radiologists and with partners at the University of Alberta Medical School, they've agreed to help recruit some of the radiology residents to participate in these experiments. But anyway, at the moment, we're starting with birds. So we're training our neural nets to categorize images in the Caltech birds data set, training them both with the labeled images and with fMRI data collected while human participants look at pictures of birds. And we can have those subjects be either expert bird watchers who are really good at telling you the kind of species each bird is, or naive controls. And we'll repeat our procedure of training the neural nets to do the task while also mimicking the brain data, using the data either from the experts or the non-experts, with the hypothesis being that the neural nets trained with the expert brain as a teacher will function better than those that are trained with the non-expert brain. And again, the longer term direction where we're taking this is not this birds task, which is fun, but not super important. But the longer term is to apply this to a more technologically important task like radiology, automated diagnostics. Yeah. So I'll turn now then to the second sort of half of my talk. We started by talking about how to use brain activity directly to teach neural nets how they should represent information. Describe to you now a bit of work ongoing in the lab where we're directly inserting neuron models into deep learning architectures in order to make those architectures function in a more brain-like manner. And I should say this is work that's really being led by Saad Idris, a postdoc in my lab. And so the idea is that we're going to look for architectures that, again, returning to this idea at the start of the talk, that the good choices for making brain-like AI are the ones that lead to systems that can predict brain activity under diverse conditions. And we're going to do this by looking for architectures that enable us to predict brain activity across diverse conditions. And the unfortunate fact that I'll show you in a moment is that the current standard CNN architectures that we thought were pretty good and probably sufficient for this, they're not, they fail. But we've come up with a scheme that I'll show you can fix this issue by basically adding new neuron-like units into our artificial neural nets. So we'll start with what we think is basically the easiest task we can think of in terms of predicting brain activity, which is one I alluded to earlier, where we predict the activity of neurons in the visual system, so either retina or visual cortex, in response to visual stimuli. And what I showed you earlier is that using standard CNNs, we were able to do this task pretty well. So we could take in pictures, predict neurons in primary visual cortex. A related paper from Surya Ganguly's group and Steve Backus is the retina experimenter on this down at Stanford, took in pictures and predict the activity of retinal ganglion cells, so the sort of output signals that travel from the eye along the optic nerve to the brain. And in both of these cases, we see that after training, taking images, predict the neuron activity, they work pretty well. What was not realized until very recently is that if you very slightly change the experimental conditions, these things fail quite catastrophically. And I'll show you how in a moment. But the first thing to note is that in basically every visual neuroscience experiment essentially ever done, the lighting conditions have been very, very carefully controlled. So you sit your animal down in a room, very carefully control the mean luminance level and the contrast, and do not vary those throughout the experiment. And if you instead look at what happens when you go out in the world, you'll notice that that carefully controlled experimental setting is very different from the way our visual system has to work. So this is data showing the optical power. It's a measure of light intensity as a function of time, while a technician from the lab of my collaborator, Greg Field, just carried an optical power meter around through a walk in the Duke University campus on a sunny day. And what you notice is that the mean light intensity varies by around a factor of 100, extremely frequently and rapidly. In hundreds of milliseconds, you see 2 log unit changes in the mean light intensity. Basically, as the person walks under the shadow of a tree branch or out from the shadow, or as cloud cover moves around, you see this enormous lighting fluctuations that again are basically ignored in visual neuroscience experiments. And so we wanted to see if our CNN architectures, basically were able to capture the way the visual system works in the presence of these more realistic conditions. And so what we did is we trained our CNN to take in, images. In this case, these are white noise checkerboard images, and the corresponding patterns of activity recorded in retinal ganglion cells. Trained our neural net using data collected at 2 light levels, in this case, 30 R star units or 3 R star. These are just measures of light intensity sort of in units of rod isomerizations per second, but basically in retina units. So we trained at 2 light levels, and then we tested using data at a 3rd light level to see whether our neural nets were able to successfully generalize across these ranges of conditions. And again, the range here, the 2 log unit range, is exactly the type that you encounter every day when you go out and wander around in the world. And if we look at the predictions for held out data at the training light levels, that's what these 2 are here. We see the neural net does extremely well. For this example cell, the neural net's able to explain 90-some% of the variance, and the median across all the neurons in our data set is 80-some% of the variance. So the neural net does extremely well at predicting neural activity in exactly the conditions in which it was trained. And at this 3rd testing light level, it's terrible, right? It's down at 24% is the median, telling us that the models do pretty well under exactly the conditions they're trained at, but they do not generalize to different conditions at all in the way the visual system does. So what we did next is we decided we were going to try to build new architectures that might fare better at this task of predicting visual neuron activity under different conditions. And we did this by just adding a new type of artificial neuron layer at the input to our deep CNN that's built around a photoreceptor model that I'll show you in a moment. And the idea is that we know that a lot of the adaptation that's changing the way the retina transforms visual stimuli into neuron spikes, we know that a lot of the adaptation is happening at the photoreceptors. That's both, quite rapid, but also quite profound in terms of how it changes visual sensitivity. And hence we hypothesize that putting in a photoreceptor-like artificial neuron might fix this problem. So to do that, we took this sort of coupled differential equation model that describes the biochemical signaling cascade within each photoreceptor. I won't trouble you with all the details of the G proteins and other things within the signaling cascade, but nonetheless, it's the thing that takes in the light, converts that into some internal chemistry in the photoreceptor that then turns it into electrical current. And this process, importantly, has both feed forward and feedback components within it. So we took a differential equation model, a couple differential equations describing the signaling cascade. This comes from the lab of collaborator Frederici. We can, backprop through this whole model to train all of the parameters of this photoreceptor model in an end-to-end fashion, along with the rest of the deep CNN. And we implemented this just as a new type of convolutional neural net layer. And when we did that, we could then train the neural net that, again, contains this photoreceptor layer in addition to conventional deep CNN layers. We could train at 2 different light levels, test at a 3rd light level, and find that at the 3rd sort of held out light level, we're able to capture about half of the variance in the neuron activity with the CNN, whereas with the sort of conventional CNN without the photoreceptor layer, we only capture a quarter of the variance at that 3rd light level. So the problem's not fully solved yet, but to us, this is very encouraging and tells us that putting in components that mimic the known physiology of the nervous system can move us further along this path to making neural nets that are able to predict brain activity and neuron activity under diverse conditions. So, to sort of loop back a bit to the way I started this talk, the idea that I'm working on is that the reasonable choices for making models that can reason in a human-like way, the reasonable choices are ones that lead to models that can predict brain activity under diverse conditions. We see even in terms of simple tasks involving vision, that the current kind of standard CNNs are insufficient for doing that. As soon as you change the light levels a little bit, they fail. But adding some new biorealistic CNN unit types can fix this problem a fair bit. So far, we've incorporated just a photoreceptor model. The next steps in this line of research are to implement a bipolar cell model and a ganglion cell model that mimic adaptation at those different stages of visual processing. And there's a whole bunch, I think, of different neuron mechanisms that are quite easy to build into deep learning architectures that I think can really, again, push us a pretty far way along this path to making models that can predict brain activity. All right, so to sort of recap the whole talk for you, we started by asking how we can make AI systems that reason in a more human-like way. Talked about a couple of different branches of research in my lab that attempt to do that. The first is work, largely led by my former PhD student, Callie Federer, where we directly use brain activity data as a representation teacher to show the neural net how to describe input data. And the second branch is where we directly use neuron models in deep learning architectures to enable them to adapt to changing conditions the same way the nervous system does. So I'll thank the people who contributed to this, especially postdoc Saad and former graduate student Callie, and then the PIs who contributed to this, and of course all the funders that enable us to do this work. Thanks very much.
Thanks, Joel. All right, so I would now invite people to ask questions. Please come to the microphones, and also you can post your questions online, and we'll get to those as well.
Hi, I'm Isabo. I'm wondering, very nice talk, by the way, really interesting. And I was curious about your first part of your presentation where you tried to mimic the monkey brain representation to achieve better classification accuracy.
Yeah.
In your slide, it seems to me that you're writing that you're only matching the V1 representation and not like the IT or something. And that's really intriguing. Is there any reason for that?
Yeah, that's a really, really good question. And exactly as you noted, we were just using monkey V1 data here, not like V2, V4, and IT. In that early study, we did also attempt to use data from V2 and V4. And for reasons we could speculate about. But anyway, the short answer is we did not find that those led to improved performance. And even when we used them in combination with the V1 data, we were unable to see the same improvements in performance in the object recognition task. Part of where we think a big issue there is that we had V1 data, V2 data, V4 data, all from different animals. And the monkey data were not collected with the same images that have category labels for the categorization task. And so, we can nonetheless train the neural net to mimic. Let's see if I can scroll back quickly to that slide. We can nonetheless, train the neural net. We basically evaluate this part of the loss on the images that the monkey saw, which we put into the neural net, and the monkey saw them. And then we evaluate this part of the loss on the category labeled images from the CFAR 100 data set. So we could do the training procedure here, but it's worth noting the images for this loss are not the ones from the same categories as the ones in the CFAR 100 data set. And that means that as you get to more categorical representation parts of the monkey brain, namely the deeper parts like V4 and on towards IT, the way these images are being represented is not necessarily as useful for categorizing the specific objects in the CFAR 100 data set. Whereas the earlier stages, namely V1, that have a less object-specific form of representation, the neural net can nonetheless show, the monkey can nonetheless show the neural net, which features to pick out that are generically useful across categories. So that's what we think is happening. But in the early studies, we did not see the same benefits for V2, V4, and IT data. Yeah, good question.
Makes sense. Thank you.
Yeah, Puya.
Hi, Joe. Thanks for the great talk. I'll just have one question again on the same slide, I guess. My question is, to what degree do you think this gain in having a loss term that tries to mimic the brain responses works in the regime that we are having a low amount of data for object recognition versus where we have a lot of data for object recognition? Like to give you an example, for example, if you were to try the same thing on a larger data set like ImageNet, would you tell us what you think would be the eventual result, or what you expect the result would be?
Yeah, so that's a really good point, right? So C5 fairly small data set. We know that if you have sufficiently large model and sufficient amount of training data, you're going to get extremely good performance, right? The state of the art for ImageNet is not quite perfect, like 100% performance, but it's getting pretty close. So there's not a lot of room left in that regime for the brain data to make a big difference. And so where we think we're going to see, in the longer term future of this project, where we think we're going to see more benefits are in the data poor regime. And where we're trying to push this now is to really go to training the neural nets in an unsupervised way using contrastive losses that can include, the images and the brain data, and let us form the image representations that are brain-like, and then similar to the way CLIP can do zero-shot categorization to move on to explore that exact question. Yeah.
Thank you. So we also have some questions from Zoom.
Yeah. Okay, we have a question from Eric Elmaznino. And the question is, could it be that the term in the loss for fitting brain data is just useful because it is a regularization term that helps prevent or overfitting? Does doing this give better performance on categorization than using something like L2 regularization?
Yeah, so the short answer is yes, this can be better than just using L2 or L1 or some of the parameter norm regularization schemes. These models, all of these we trained using dropout, and we had titrated the dropout sort of keep rate to try to get good performance. And nonetheless, we see that adding the brain data makes things better. So there is room beyond the sort of standard suite of regularizers for brain data to make some difference in performance.
Actually, I have a question.
Yeah, go ahead.
Yeah, so you describe how you can use brain data and recognition performance to constrain the network. But what about doing constraining it by specific experiments that have been done? For example, people have looked at how luminance affects activity in V1 and how it affects performance, and they build all kinds of contrast curves, etc. Could you use the results of those experiments? I mean, there's decades of experiments to further test the network and add some other sort of loss if it doesn't fit the experiments, from the 70s and 80s, both at the behavioral level as well as at the neural level, because there's many ways of getting good performance or getting good similarity, representational similarity metrics, but the brain has a particular one with particular nuances. So have you included that in some of your models?
Yeah. Yeah, those are great questions. The short answer to your question is no. We have not included as sort of added regularizers all of the different behavioral data or neuron data from all the different, say, luminance conditions that we could sample. I think that's an excellent idea. One quick pitch: there is a nice platform that some friends and colleagues of mine have developed called Brain Score, that does take deep learning models and evaluate their brain similarity in terms of both the neuron activity data and behavioral data. So there are benchmarks being developed, not by me, I should say, but by Koetish Carr and Martin Shrimp and Tiago Marques from Jim DiCarlo's group. Again, Brain Score is that platform that are pushing in that direction, but we haven't done that yet.
Okay, I have a question from Alexandre Payer. which is representational similarity is only a self-consistent statement about category representation.
Agreed.
It does not tell whether neural representations themselves are similar. Does
Yep.
deep CCA correct for this?
Yeah, so exactly. So this is exactly one of the motivations to moving to deep CCA is that, you know, the RSM is really limited. And in fact, one of the limitations is that we have a lot of units in the artificial neural net that we're comparing to about 100. So a pretty small number of neurons that we can sample from the monkey brain. And if we force that whole big neural net layers RSM to match the small number of neurons from the monkey brain, we're really forcing it to restrict the set of operations that are done or the set of features detected by this large group of units, because again, they have to match this small number of neurons from the monkey brain. So one of the benefits to deep CCA is that it basically, you know, takes in the monkey data or the unit activations from the neural net, and then takes a deep artificial neural network, not extremely deep, a few layers deep artificial neural net, and just translates those into vectors that are then trained to be as similar as possible. And so both these weights and the weights within the CNN are trained so as to, you know, make these vectors as similar as possible. And it means that the CCA readouts can learn to, you know, just pick out, for example, the units in the CNN that should be aligned to these units that we've happened to sample from the monkey brain and can deal with that subsets problem. Can also, you know, as the question noted, can also help deal with other limitations of the RSM approach. So we're excited to be incorporating what we think is this more flexible, well, we know it's more flexible, but we think it's a better scheme for doing the brain matching. Yeah. And again, that's the topic of this paper from Cassidy Pierlot is the lead author.
Thank you for the great talk. So I was wondering, did you actually look at what changes in the neural network when you constrain the brain? Is it like distributed over the whole filters, or is it like there's some certain pathways that have profound changes?
Yeah, that's a good question. So so what's actually changing in the neural net when it's forced to match the brain? We looked quantitatively, partly just to debug the things and make sure it worked. We looked quantitatively at whether the brain similarity actually improved when you train the neural net to mimic the brain. It does. Maybe that's not surprising. We did not look in detail though, at like the shapes of the kernels in the neural nets trained under those different regimes, and that's a good idea. But we did not do that.
Thank you for the wonderful talk. I'm Chen. So I was thinking one feature of the brain is the variability. So it's variability as a function of trial and variability across individuals. Is there a way to integrate that into the neural net?
Yeah, so is there a way to incorporate neural variability into neural nets? Sort of. So, I mean, we kind of introduce some of it in the current schemes with things like dropout and drop connect as regularizers to help training. So there are some ways in which the variability seen in the nervous system can be directly incorporated. What we think is going on, especially to go back to this retina project, some of the variability we think happens just because of hysteresis. So if you show me the same picture a few times and record neuron activity, you'll see different firing patterns in retina and in visual cortex. Especially in retina, some of that difference is probably because the preceding stimuli, the pictures I saw before the one that we're looking at the neural activity for, were different, and they differentially adapt the different parts of the retina so that you're effectively showing that same picture to different retinas each time. And so one way we can help address that is by having sort of these neural net layers that adapt the same way the retina does. That means that the response, the sort of output of this network to the same picture will differ depending on what happened before, but will do so in the same way as the retina. So there are some ways in which we're incorporating neuron variability into artificial neural nets, but it's by no means the full picture.
Right. Well, please join me in thanking Bill.